What if I told you that editing life's instruction manual will soon feel as ordinary as sending a text? Yesterday's sci-fi, reprogramming our own DNA, has burst out of the lab and into the driver's seat of humanity's future. Welcome to an age where life itself is programmable. Imagine walking into a grocery store decades from now. You scan a drought-proof tomato, grab coffee beans grown in carbon-absorbing soil, then pick up a pill engineered to reboot your immune system against cancer. That isn't fantasy. It's happening now. We've gone from blasting seeds with radiation and praying for lucky mutations to wielding CRISPR like a molecular scalpel, snipping and swapping DNA in days. In the 1960s and 70s, pioneers tinkered with bacteria and mice, laying the groundwork for today's biotech. By the 80s, engineered microbes cleaned oil spills and churned out human insulin without harvesting animal organs. The 90s brought us the flavor-saver tomato and 3-parent IVF babies. Yet all of those leaps were clumsy compared to the CRISPR revolution. Suddenly, gene editing costs plummeted by 99%, and timelines shrank from years to mere weeks. CRISPR co-opts a bacterial defense system. Microbes archive snippets of viral DNA, craft a guide RNA, and deploy a Cas9 search and destroy team to edit virtually any gene with pinpoint accuracy. Labs from Boston to Bangalore are curing genetic diseases, eradicating crop pests, and even dreaming of resurrecting extinct species. The real-world impact is miraculous. In 2015, scientists snipped HIV out of infected human cells in the lab. A single CRISPR injection erased over half the virus from rats. Clinical trials are already unleashing CRISPR-enhanced T cells against tumors. Over 3,000 genetic disorders caused by single-letter typos in our DNA, cystic fibrosis, sickle cell anemia, muscular dystrophy, stand within striking distance of a cure. But such power demands responsibility. DIY biohacker spaces are sprouting in garages worldwide, democratizing access and raising biosecurity alarms. That's why researchers build molecular kill switches into CRISPR systems, publish open-source safety protocols, and form international consortia to share data. The same kit that grows glow-in-the-dark yeast could, if misused, resurrect pathogens we once defeated. Equity is our next front. Will life-saving therapies be priced for the wealthy? Or can we learn from past mistakes? Tiered pricing, non-profit partnerships, open-source vectors to ensure global access. Imagine gene drives wiping out malaria vectors in sub-Saharan Africa, but only if local communities guide deployment and consent to trials. The decisions we make now will define who benefits and who's left behind. Beyond medicine, gene editing could remake humanity. Once we master safe embryo edits, will we stop at preventing deadly diseases? Or will parents opt for perfect vision, enhanced strength, or even a boost in cognition? Pioneer labs in China already experimented with human embryos in 2015 to 16, revealing both promise and peril. Designer traits could become the new normal, sparking debates about diversity, fairness, and societal pressure. Environmental applications are equally staggering. We could engineer drought-resistant crops feeding millions, heat-tolerant corals reviving dying reefs, and trees modified to absorb more carbon to combat climate change. Yet releasing engineered life into complex ecosystems demands humility. How will these organisms interact with nature? Ecologists, indigenous communities, and policymakers must collaborate to chart safe paths. Looking ahead, next-generation editors like base editors and prime editors promise surgical precision, rewriting single DNA letters without cutting the double helix. Epigenetic editors could dial genes up or down without altering the code at all. We might one day reboot extinct species, woolly mammoths grazing the Arctic or passenger pigeons reclaiming our skies, blurring conservation and creation. Pair gene editing with AI-driven design, and the possibilities explode. Algorithms could predict the perfect gene variant for any environment, then automate lab work in high-throughput biofoundries. Custom organisms for medicine, agriculture, or environmental repair could roll off assembly lines like software updates. The boundary between code and life will vanish as biology becomes another programmable medium. All this unfolds against a changing planet. Pandemics, food insecurity, biodiversity loss, aging populations. Gene editing offers tools: rapid vaccine development, personalized cancer therapies, anti-aging interventions. Yet it poses profound ethical dilemmas. Who decides which traits are desirable? And what safeguards protect those unable or unwilling to edit their genomes? Public policy will shape the future nearly as much as science. Nations will compete for biotech leadership, setting divergent standards, from life-saving therapies only to outright designer baby regimes. We must ask ourselves, what kind of world do we want? One of uniform optimization, or one that cherishes diversity, individual choice, and the right to remain unedited? Your voice matters in this grand experiment. Stay informed, read beyond the headlines, and support policies that balance innovation with caution. Encourage your representatives to fund ethical research, craft thoughtful regulations, and foster global collaboration. Demand transparency from companies and scientists, and engage in community dialogues about the values we hold dear. We stand at the dawn of our species' greatest rewrite. The choices we make, how we govern gene editing, who gains access, and which projects earn public trust will echo for centuries. Will we use CRISPR to heal, protect, and explore? Or will unintended consequences reshape life in ways we can't yet predict? If this glimpse into our genetic future fired you up, smash that like, subscribe, and ring the bell for more deep dives into the science shaping tomorrow. Share your thoughts below. Which CRISPR breakthrough excites you most? Or which ethical question keeps you awake at night? Together, we'll navigate this uncharted territory and write the next chapter of life itself.